Hello everyone. In this lecture, I will talk about the histology of the lymphoid tissue, in particular the histology of the lymph node. At the end of this lecture, students should be able to identify the lymph node under the microscope. They should be able to describe the microanatomy of the lymph node. They should be able to correlate the structure of a lymph node with its functions. The immune system performs the role of defense of the body against infectious and non-infectious foreign substances. The lymphatic system is the morphologic counterpart of the immune system. It comprises of all the cells, tissues, and organs that are involved in the defense mechanisms of the human body. The lymphatic system is intimately associated with the blood vascular system, and together these two constitute the circulatory system. Now, most of the tissue fluid that enters the capillary bed through the arterial system, it escapes into the extracellular compartment, and from here it re-enters the venous system and comes back to the heart. However, some of the tissue fluid is returned to the circulation through a separate system of lymphatic channels. This system of lymphatic channels begins in the form of lymphatic capillaries within this capillary bed, and some tissue fluid enters these lymphatic capillaries. From here it will pass to the lymphatic vessels and then to the larger lymphatic trunks, lymphatic duct, and is finally returned to the venous circulation. The lymphoid tissue. The lymphoid tissue comprises of cells and the lymphoid organs. Amongst the cells of the immune system, there are the T lymphocytes that participate in cellular immunity, the B lymphocytes that take part in the humoral immunity, and the natural killer cells that are cytotoxic against several cancers and the viral infected cells. Now, the B lymphocytes, they recognize the antigen, and when they get stimulated, they produce antibodies that will neutralize the pathogens, phagocytose the pathogens, and also activate the complement pathway. The T cells, the T lymphocytes are further of several types: the helper T cells, cytotoxic T cells, and regulatory or suppressor T cells. Helper T cells perform the function of activation of macrophages, activation of other T and B cells, and take part in the inflammation. The cytotoxic T cells, when they recognize the antigen presented by the other cells, they take part in elimination of the infected or the malignant cells. Whereas the suppressor T cell or the regulatory T cell performs the function of regulation and or suppression of the immune response. So this cell acts as a check mechanism. The natural killer cells also take part in elimination of the infected or the malignant cells. Coming to the next component of the lymphoid tissue, that is the lymphoid organs. They can be classified into primary lymphoid organs and secondary lymphoid organs. Primary lymphoid organs in the human body are the thymus and the bone marrow. These are the sites where lymphocytes are actually produced and they undergo maturation so that they become capable of recognizing antigens. The secondary lymphoid organs include the spleen, the lymph nodes, and the mucosa-associated lymphoid tissue such as the Pierce patches in the intestines, appendix, and the tonsils. In these organs, the mature lymphocytes from the primary lymphoid organs will migrate, and here, when they get exposed to an antigen, the lymphocytes get activated and become immunocompetent B and T cells and thus initiate an immune response. The lymphoid tissue can also be classified into the diffuse and dense lymphoid tissue. Both of these guard the body and are the sites of initial immune response. The diffuse lymphoid tissue. In this type of tissue, the lymphocytes and other free cells are found in the lamina propria of the mucosa. This type of diffuse lymphoid tissue is seen in the alimentary canal, respiratory passages, and the genitourinary system. It is also known as the MALT, the mucosa-associated lymphoid tissue, because of its location in the lamina propria of the mucosa. So here you can see the lamina propria of the gut. This is the entire mucosa, this one being the epithelium, and underneath it is the lamina propria, and you can see that in the lamina propria several lymphocytes are diffusely scattered throughout, and these constitute the mucosa associated lymphoid tissue. The presence of these cells just close to the lining epithelium is a very strategic location whereby the lymphocytes are able to intercept the antigen and initiate an immune response. When the lymphocytes present here will get in contact with any antigen that comes in touch with the lining of the gastrointestinal tract, these cells, they will travel to the regional lymph nodes, they will proliferate and differentiate, and the progeny of those lymphocytes will return back here to the lamina propria as effector B and T lymphocytes, and they will be able to counter and destroy the antigen that has come in contact with the lining of the mucosa of this part of the gastrointestinal tract. So this is the diffuse lymphoid tissue. Now the dense lymphoid tissue is seen in the form of lymphatic nodules. It can be found either as discrete encapsulated organs, or it can be seen in association with the epithelium of certain regions. The discrete encapsulated organs include the thymus, lymph node, spleen, and tonsil. Whereas the sites where it is associated with the epithelium include the mucosa-associated lymphoid tissue. It is also further of several types. For example, the bronchus-associated lymphoid tissue, also called BALT. Here the lymphatic nodules are seen in intimate association with the lining epithelium of the trachea and the larger bronchi. Then there is the GALT, the gut-associated lymphoid tissue, where lymphatic nodules are seen in the wall of several regions of the gastrointestinal tract, the prominent among them being the Pierce patches in the intestines, the adenoids in the nasopharynx, the lingual and palatine tonsils in association with the oral cavity, and also the appendix which is a part of the large intestine. Some of the mucosa-associated lymphoid tissue is also seen in association with the female reproductive tract, where the mucosa is exposed to the external environment. Let us now discuss about the lymph node. Lymph nodes are bean-shaped masses of lymphoid tissue along the course of blood vessels. These serve as filters through which the lymph percolates on its way to the blood vascular system. Lymph nodes are widely distributed in the body, and they are especially concentrated in certain regions like the axilla, the groin, the region of the neck, and the mesenteries in the gastrointestinal tract. The basic structure of a lymph node comprises of a supporting framework, the parenchyma, and the sinuses. The supporting framework of the lymph node includes the outer capsule and then the extensions of the capsule into the parenchyma, which are called the trabeculae, and the reticular tissue framework within the parenchyma of the lymph node. The entire parenchyma of the lymph node further is divided into an outer cortex and the inner medulla. Underneath the capsule of the lymph node, there is the presence of a subcapsular sinus. These subcapsular sinuses continue with the sinuses around the trabeculae, which are called the trabecular sinuses, and these trabecular sinuses further continue into the sinuses of the region of medulla, which are called the medullary sinuses. So this is the basic structure of a lymph node. This is a panoramic view of a lymph node in hematoxylin and eosin stain at a low magnification. You can appreciate the presence of a capsule all around the lymph node, and within this capsule you can also appreciate the afferent lymphatic vessels. This is the parenchyma of the lymph node, and these are the extensions of the capsule into the parenchyma of the lymph node. These extensions are called the trabeculae. Within the parenchyma of lymph node, there is an outer cortex and an inner medulla. The outer cortex is studded with several lymphatic nodules. You have to understand why this cortex is appearing as if containing so many bluish basophilic dots. For this, you have to understand the structure of a lymphocyte. The lymphocytes have got a large nucleus which is very densely stained, and this nucleus occupies most of the cytoplasm of the lymphocyte. In fact, the cytoplasm is reduced to a very thin basophilic rim around the nucleus. So, at low magnification, you see the lymphatic nodules as containing several bluish lymphocytes, which are actually the nuclei of the lymphocytes that you are seeing here in low magnification. So the cortex is studded with these lymphatic nodules, and this is the region of the medulla where there are several medullary sinuses that you see as the pale staining regions here, and around the sinuses you can see again the basophilic dots here, which are nothing but the lymphocytes arranged as cords around the medullary sinuses. Also, a very characteristic feature of the lymph node is this subcapsular sinus, which is a pale staining region present just underneath the capsule of the lymph node. This subcapsular sinus, which is a very characteristic feature of lymph node, is lined by endothelium and it receives the lymph from these afferent lymphatic vessels. Now, this is the region of the hilum of the lymph node. You know that the afferent lymphatic vessels enter the lymph node through this convex region by piercing the capsule, and the efferent lymphatic vessels will exit the lymph node at the region of the hilum. The hilum will also contain the arteries and veins that give the blood supply to the lymph node. The supporting framework of the lymph node. The capsule is the outermost covering of the lymph node, and it is made up of dense, irregular connective tissue. Now, in this capsule, you can see numerous collagen fibers along with the nuclei of the fibroblasts. These fibroblast cells had actually secreted this connective tissue capsule. You can also see the afferent lymphatic vessels within the capsule. Now, just underneath the capsule, there is a slightly pale staining area, which is the subcapsular sinus. This sinus is lined by endothelium, and the afferent lymphatic vessels, they pierce the capsule and they drain their lymph containing the lymphocytes into this region of the subcapsular sinus. Now, the capsule sends extensions of connective tissue into the parenchyma of the lymph node, and these are called the trabeculae. So trabeculae are also made up of dense irregular connective tissue. They contain the major blood vessels of the lymph node. The trabeculae are surrounded by the trabecular sinuses, and whatever lymph is drained by these afferent lymphatic vessels into the subcapsular sinus further travels in the trabecular sinuses. Now, the reticular tissue framework, which is seen in the parenchyma of the lymph node. Now, this is a view of the lymph node at high magnification in silver stain, and here you can appreciate the fine network of the reticular fibers, which are the supporting framework for the lymphocytes and the other cells within the parenchyma of the lymph node. This reticular tissue framework comprises of the reticular cells that you see here. These reticular cells secrete the reticular fibers, which are stained here with the silver stain. This meshwork of reticular fibers contains several cells apart from the reticular cells. These include the macrophages, dendritic cells, and the follicular dendritic cells. The reticular cells secrete type 3 collagen and the ground substance of the stroma of the lymph node, and their processes wrap around the reticular fibers. These cells are special and they secrete some surface molecules to attract the lymphocytes and the dendritic cells. The macrophages present here are actually the antigen presenting cells, and they are also phagocytic in nature. Now let's discuss about the parenchyma of the lymph node. The parenchyma of the lymph node, I have already told you, is divisible into an outer cortex and the inner medulla. Now the cortex is the outer portion of the lymph node. It is further divided into outer cortex and a deep cortex, also called the paracortex. The outer cortex forms the outer portion of the lymph node. It comprises of dense masses of lymphatic tissue which are called the lymphatic nodules. These lymphatic nodules are basically aggregates of B lymphocytes. These lymphatic nodules are of two types, primary and secondary lymphatic nodules. Here in this hematoxylin and eosin stained picture of a lymph node, you can appreciate that this is a primary lymphatic nodule. The primary lymphatic nodule comprises of small lymphocytes which are uniformly scattered across the lymphatic nodule. However, in case of a secondary lymphatic nodule, you will see a pale staining region in the center, which is called the germinal center, and the outer darker staining region, which is the mantle zone or the corona. Now this picture is showing you the outer cortex of the lymph node and also the paracortex over here. This is a medium magnification picture under the hematoxylin and eosin stain, and here you can appreciate a large secondary lymphatic nodule. Let us discuss the structure of a secondary lymphatic nodule in more detail. Here in the center, there is a relatively pale staining zone which is called the germinal center. Now this region appears to be pale staining because here the nuclei of the lymphocytes are relatively lighter in appearance. This is because this region comprises of actively proliferating cells. In fact, this germinal center develops when there has been an exposure of the B cells to an antigen. So when the B cell has recognized an antigen, it starts to undergo a lot of proliferation, and several actively dividing lymphoblasts and plasmablasts are seen in the central portion. These cells have got a lighter staining euchromatic nucleus because the chromatin is less condensed. These are large rounded cells with a broad band of cytoplasm and a large vesicular nucleus with one or more prominent nucleoli. These actively dividing lymphoblasts will give rise to small lymphocytes. Now these small lymphocytes will aggregate in the peripheral zone of the secondary lymphatic nodule, and these small lymphocytes have darker staining condensed chromatin containing nuclei. That is why this zone appears dark, and this zone is called the mantle zone or the corona. So this is about the secondary lymphatic nodule. Further deep to the outer cortex is the region of the paracortex. This deeper zone of the cortex comprises of T lymphocytes. That is why this zone is also called thymus dependent cortex. Coming to the medulla comprises of the medullary cords and the medullary sinuses. Also, there will be the reticular tissue framework supporting these cords and sinuses. The cells that are seen in the medulla are mostly B lymphocytes, macrophages, dendritic cells, and plasma cells. So, please note that in the cortex, in the outer cortex, there were mostly B lymphocytes in the lymphatic nodules. The paracortex contains mostly T lymphocytes, and the medulla contains mostly B lymphocytes. These cells are arranged in the medulla in the form of anastomosing or branching cords, and the adjacent cords are separated by the medullary sinuses. These medullary sinuses are lined by endothelium. So the walls of these sinuses will be lined by flattened squamous cells. All these medullary sinuses will finally converge towards the region of the hilum of the lymph node, where they drain off into the efferent lymphatic vessels. Let us now discuss about the sinuses found in the lymph node. Just underneath this connective tissue capsule, there is this pale staining area which is called the subcapsular sinus. It separates the cortex from the capsule. This sinus is lined by endothelium containing the flat endothelial cells, and this sinus is continuous with the endothelium lined sinuses present around the trabeculae, which is the trabecular sinuses. Now you can appreciate the endothelium of the sinus here. These are the flattened nuclei of these endothelial cells. You can see one cell here and one cell here. So please remember to incorporate the endothelium with the flattened nuclei when you draw the diagram of the lymph node and when you draw the subcapsular and trabecular sinuses. Now from here, from the trabecular sinuses, the lymph will finally enter into the medullary sinuses in the medulla. Now, I told you that these sinuses are lined by endothelium. The walls of these sinuses, they are continuous where they line the capsule and the trabeculae. However, their wall is discontinuous towards the side where the sinuses face the parenchyma of the lymph node. And you can see a discontinuity here. And the lymphocytes and macrophages or their processes can readily pass back and forth between the sinuses and the parenchyma. In this manner, the lymphocytes from the lymph flowing in the sinuses and the macrophages can enter into the parenchyma of the lymph node. Now there is a special entity that is found in the lymph nodes. These are the high endothelial venules. Let us see what are these high endothelial venules. I told you that the lymphocytes traveling in the lymph coming from the afferent lymphatic vessels can escape across the walls of these sinuses into the parenchyma. However, most of the lymphocytes that enter the lymph node, roughly 90% of the lymphocytes that enter the parenchyma of lymph node, they enter the lymph node through the walls of the post-capillary venules that are located in the deep portions of the cortex, that is the paracortex. So in the paracortex, the post-capillary venules that are present, they do not have the typical endothelium that is seen in the venules. These high endothelial venules, they are lined by tall cuboidal or columnar endothelium. You can appreciate a high endothelial venule in the paracortex in this H&E picture at high magnification, and you can appreciate the rounded nuclei of the tall cuboidal epithelial cells, endothelial cells that are lining this high endothelial venule. A special property of this tall cuboidal endothelium is that it can secrete chemoattractants for T and B lymphocytes. And because of this, the T and B lymphocytes can migrate across the high endothelial venule into the parenchyma of the lymph node. This process by which the lymphocytes they escape out into the parenchyma of lymph node is called homing or diapedesis, that is similar to that done by the neutrophils in other regions of the body. So the parenchyma of lymph node is receiving majority of the lymphocytes from the migration across these high endothelial venules, and some lymphocytes are also entering across the walls of the sinuses. Let us now summarize the circulation of lymph through the lymph node. We will begin with the afferent lymphatic vessel. These vessels have got valves in them, and these vessels pierce the capsule of the lymph node and drain the lymph into the subcapsular sinus. From here, the lymph will travel in the trabecular sinuses and then to the medullary sinuses, and finally the lymph will exit the lymph node at the hilum into the efferent lymphatic vessel. So this is how the lymph will circulate through the lymph node. The efferent lymphatic vessel will further take the lymph to some other lymph nodes or to the other larger lymphatic vessels, lymphatic trunk, and finally the lymphatic ducts, and the lymph goes into the thoracic duct through which it goes into the left subclavian vein and then back to the heart. You have to note that when the lymph reaches the region of the medullary sinuses, the B cells have released antibodies into this lymph to counter the antigens that are harmful to the body. So that means from the medullary sinuses onwards, the lymph flowing through the lymph node will be very rich in antibodies which are distributed to the entire body through the general circulation. Coming to the blood supply of the lymph node, the arteries enter the lymph node through the hilum. They traverse through the medulla and they break into smaller arteries that reach the cortex, where they break into the capillaries, and then the venules, the post-capillary venules or the high endothelial venules in the paracortex, and finally they exit the lymph node through the efferent vessels or the veins, which will leave at the region of the hilum of the lymph node. Functions of the lymph node. Lymph node is the center for lymphocyte production. Antigens are conveyed through the lymph and they initiate an immune response. And when antigen exposure happens, there is activation of the T and B lymphocytes. Just one word about the B lymphocyte differentiation. When the B lymphocyte gets activated by an antigen, this naive B lymphocyte gets activated and it will actively divide and it will become a lymphoblast, also called here the plasmablast. This plasmablast will migrate to the bone marrow and they will keep producing antibodies for prolonged periods to counter the antigen that has come into the body. When these lymphoblasts divide, some of them become the memory B cells. These memory B cells will leave the lymph node and they will circulate to various regions of the body where they can proliferate and generate an immune response whenever there is a subsequent or a future exposure to the same antigen. Because of this, they are called the memory B cells because they still have the memory of exposure to that antigen. The presence of memory B cells in a widespread fashion across the body ensures a more rapid secondary response to an antigen. So some of the lymphoblasts had gotten converted into the memory B cells, and the others, they form the plasma cells. These plasma cells will migrate to the region of the medullary cords and they release the antigen into the lymph. And that is why I told you that the lymph flowing from the medullary sinuses onwards is very rich in antibodies that will be distributed to the entire body to fight that antigen. Coming to the clinical aspects, first of all, lymphadenitis. Lymphadenitis refers to an enlarged lymph node due to an infection. You can see here that the lymph nodes in the neck have gotten enlarged because of exposure to some bacteria or virus or any other infection. Lymphangitis is the inflammation of the lymphatic vessels. You can appreciate the inflamed reddish lymphatic vessels. This is the lymphangitis. Now, a word about the spread of cancer across the lymph nodes. The lymph nodes are capable of trapping antigenic material and malignant cells reaching the parenchyma of the lymph node through the sinuses. However, if the cell load is excessive, this filtration system can get overwhelmed by the increased number of cancer cells flowing through the system. As a result, cancer cells may get established within the lymph node and develop a new metastatic site within the lymph node. So, you can see here in this picture that this was the tumor, and the malignant cells, they travel through the lymph and reach the lymph node, and sometimes they establish a metastatic center within the lymph node itself. Also, some of the cancer cells or malignant cells will escape. They will pass across the lymph node and thereby they can get distributed to the other distant lymph nodes, or they can reach the general circulation and thereby establish metastatic sites in the distant organs. So this is how the lymph node itself can become a metastatic site for cancer, and the lymph node can also lead to the spread of the cancer. Thank you.